Alright, hi guys. So we are doing week 4, the cell model. And so it's this mess of a model here. So our first term is the plasma lemma or plasma membrane. So that's going to be this outside portion. Remember that is a phospholipid bilayer. And so it has a hydrophobic and a hydrophilic side. Then you have the cytosol, which is the fluid component in here, so it's just going to be the space. The cytoplasm is everything inside. The cytosol is just the liquid part of it. Then you have the cytoskeleton, which really the best... Ooh, what a bad sound. The best place to see that are all of these little scratches in here. They're trying to show you the cytoskeleton, which is microtubules and different filaments and stuff like that, which you can look up when you look in your lecture. Next is microvilli, which you cannot see on this model, but if you did, there would be little protrusions of plasma lemma right here. And that function is to increase the surface area. Microvilli do not move. They are only for the increase of surface area. So you'll see them in places that there's a lot of absorption going on, such as your small intestines. The next is the centrosome with centrioles. So this is this little black thing. The centrosome is the whole thing. The centrioles, there's each of these, they participate in mitosis. They help pull apart the chromosomes. Then you have the mitochondria, which are these little orange dudes, and they are what you learned in 8th grade was probably the powerhouse of the cell, but what we're going to say now is cellular respiration, so they produce ATP. The next one is the nuclear envelope with the pores. So it's this white part with the holes in them. So the pores allow messenger RNA to leave and tRNA and such. Then you have the nucleus is the whole thing. Right? Inside you have chromatin, which are these little blue guys in here. They're trying to show you that. Chromatin is the loose form of DNA. So that means that it's active, it's able to produce or to be transcribed into mRNA and then produce proteins. Then you have the nucleolus is this dark spot right here. It stains very, very dark because it's full of histones and ribosomes and RNA. Then you have the endoplasmic reticulum. You have two types. You have smooth and rough. The rough is this part right here with all these little white guys on it. The smooth is this blue one over here without the rough on it. The rough endoplasmic reticulum is rough because of the ribosomes, and so its function is to produce proteins and also do synthesis of other cellular components. The smooth ER is there for the detoxification of the cell and also the synthesis of biomolecules such as lipids. Then you have the Golgi Apparatus, which is this pink guy up here. You can always tell because it has little vesicles along the sides. Its function is to modify, package, and ship things that need to go in and out of the cell. They don't necessarily have to be sent outside of the cell. They could be sent within, but usually when we're talking about this, we're talking about it going outside. Then you have lysosomes and peroxisomes. Now, the thing about this is it's these two things. Looking at them, the only difference is the color. So the teachers have just decided which one's which. The only way you'd really be able to tell the difference is by doing an analysis of the enzymes inside, but the ones that we have assigned is this is the lysosome, and so these are going to do apoptosis or cell death, and also it's going to digest bacteria and old organelles and the like. This guy up here is a peroxisome, and he does a very special function of breaking down hydrogen peroxide into water and oxygen, which are two things we are, you know, very okay with. That is the overview of this model. Really, there's a lot of lecture components in this, so I urge you to make sure you go over these things in your book.